Hong Li, thank you so much for the invitation and for organizing this lecture. I'm excited to be here. I wish I could be in person. I missed that part about giving talks. I'm excited to share the work that we've been doing in my research group, and I apologize for my voice. I picked up my kid's cold, so hopefully I will make it through without losing it completely. But the title of my talk today is Air Pollution and Breast Cancer, Understanding Exposure Mixtures and Underlying Pathways. So I'm going to start with a brief background on breast cancer epidemiology, just so we're all on the same page. Breast cancer is the most commonly diagnosed cancer among women in the US and worldwide. In 2021, it's estimated there were over 200,000 new US cases. Breast cancer is a heterogeneous disease, so both risk factors and survival vary by menopausal status at diagnosis, as well as tumor subtypes, which we often define using the estrogen receptor and the progesterone receptor. There are a number of established risk factors for breast cancer, and you can see that many of these tend to have modest effect sizes. So that's what we're used to seeing in breast cancer epidemiology. Some of the established risk factors for breast cancer include a woman's reproductive history. So having an earlier age at menarche, a later age at first full-term birth, or a later age at menopause. And so these all support that hormones play an important role in breast cancer etiology and suggest a potential role for endocrine disruption. Additionally, we've long known that high-dose radiation plays a role in breast cancer risk, which supports a more global role for the environment. So my work focuses on air pollution, which is a potential environmental risk factor for breast cancer. Air pollution is extremely heterogeneous, and it really varies based on these different sources that it arises from, including traffic, industrial sources, indoor heating and cooking, as well as natural events such as wildfires. And air pollution may be plausibly relevant for breast cancer risk. We know that air pollution is a carcinogen, and the International Agency for Research on Cancer has classified outdoor air pollution as a group 1 carcinogen, although this is primarily based on what we know about fine particulate matter and lung cancer. We don't typically think of air pollution as being relevant for the breast, but studies of cigarette smoking show that inhaled toxicants have been found in the breast fluid, suggesting that the compounds we inhale enter our circulation and can reach the breast tissue. And air pollution does contain this heterogeneous mixture of compounds that have both carcinogenic as well as endocrine disrupting effects, which may be particularly relevant for breast cancer, given what we know about the role of hormones in the disease. And some of these compounds of interest include polycyclic aromatic hydrocarbons, or PHs, volatile organic compounds, as well as metals. So I'm going to start by talking about a few of the studies I've done in the sister study, considering different types of air pollution in relation to breast cancer risk. So to start, to give you some background on the sister study, so some of you may be familiar with this cohort, but it is a large prospective cohort study of over 50,000 women living across the US. And so they were recruited between 2003 and 2009, at which point all the women in our study were breast cancer free. But importantly, they all have a sister, either half or full, who've been previously diagnosed with breast cancer. And so that puts our participants at about 2 times higher risk of developing breast cancer themselves. At baseline, women completed an extensive questionnaire, including on their residential history, as well as other known and suspected breast cancer risk factors. The cohort is an active follow-up, so we have women completing annual health updates, as well as more detailed follow-up surveys about every 2 to 3 years. This is a really engaged study population, which is understandable given that these women volunteered to be in the study and they all have a family history. So our response rates tend to be around 90% over follow-up. If women are diagnosed with breast cancer, they report that back to the study, either on these annual health updates, or they can call in, there's a hotline to call. And then we request access to their medical records and pathology reports to confirm their diagnosis and to get information about the tumor. So as of the most recent data released in the sister study, we have on average about 10 years of follow-up in the cohort. And during this time, we've had almost 4,000 cases. These cases are largely invasive compared to DCIS, which is considered stage 0 disease. And they're largely hormone receptor positive, which is consistent with the breakdown of tumor subtypes in the US overall. So at baseline, the average age of the sister study participants was about 55 years. The cohort is 84% non-Hispanic white and about 9% non-Hispanic Black or African American. This is a slightly higher SES cohort, with about half of the women in our study having a bachelor's degree or higher, and a third reporting an annual household income greater than $100,000 a year. So for the first thing I'm going to talk about today, we focus on the association between airborne metals in relation to breast cancer risk. And airborne metals belong to a group of pollutants called the hazardous air toxics. And these are pollutants that are expected by the EPA to have adverse health effects due to what we know about their chemical and the compounds in vitro and in vivo. And I decided to focus on metals out of all of these hazardous air toxics because certain metals have been classified as probable carcinogens, as well as the metalloestrogen hypothesis, which is this idea that certain metals have the ability to activate the estrogen receptor. In fact, evidence suggests that certain metals may be able to bind directly to ER alpha. And this results in increased proliferation of estrogen-dependent breast cancer cells, as well as increased expression of estrogen-regulated genes. So metals appear to be acting both as a potential carcinogen as well as an endocrine disruptor. And finally, studies have shown that metals have been measured in the breast tissue, suggesting that they are able to reach the breast. So to do this work, I used data from the National Air Toxics Assessment database, which is maintained by the EPA, and it provides nationwide census-tract levels of air toxic pollutants, including the metals. And this database relies on inputs from the National Emissions Inventory, as well as exposure models to estimate these levels at the census tract. And so our aim of our study was to evaluate the association between airborne metals, considering them individually and as a mixture, at study baseline in relation to breast cancer risk. And we focused on these 10 metals that are listed here. So we took the sister study address at enrollment that was geocoded and linked to the 2005 NATA estimates of these airborne metals. And then we followed women for breast cancer over an average of 7.4 years of follow-up. And during this time, we had over 2,500 cases diagnosed. So we first looked at these airborne metals individually in relation to breast cancer risk. And we didn't see evidence of associations with overall breast cancer or by ER tumor subtype. But we did see suggested evidence of a higher risk for certain metals in relation to postmenopausal breast cancer. And this graph showed hazard ratios and 95% confidence intervals. And so we saw that women who lived in areas of higher exposure to cadmium, mercury, and lead had suggestively higher risk of postmenopausal breast cancer. But we know that women aren't exposed to just one of these metals at a time, and that these metals are correlated due to the fact that they often arise from similar sources. And so I also undertook a mixtures approach called weighted quantile sum. And weighted quantile sum gets at the question of the overall mixture effect. So if we increase all components of your mixture, so in this case, the airborne metals, by 1 quintile, what's the resulting impact on your outcome? And so when we applied weighted quantile sum, we found a 10% higher risk for postmenopausal breast cancer. And so that doesn't sound like a very large effect, but it is just for 1 quintile increase. So when you extrapolate that out, comparing women in the highest quintile of airborne metals to the lowest, it translates to about a 45% higher risk.
And Lexi, can I ask a question? Sorry for the interruption.
No, that's fine.
So the exposure assessment, this was based on the address from baseline until 2000, counting for the moving status.
So this is not accounting for moving. So we just focused on where they lived at baseline and then followed for breast cancer.
And the cancer incident cancer after the exposure.
Yes. Yeah, so the women could develop breast cancer at any point after follow-up. And so we had, for the study, I think on average about 7 years between when we looked at this, assessed their airborne metal exposure at the residence.
Thank you.
Yep. So this weighted quantile sum method, it gives you this estimate of the overall mixture effect. And it also identifies those components of the mixture that are most strongly driving the association. And so consistent with our individual metal results, we found that the overall mixture was also driven by exposure to cadmium, lead, mercury, as well as cobalt. So this was the first study to consider airborne metal mixtures in relation to breast cancer risk. Some previous studies, including one led by investigators at the California Teacher Study, which is a large cohort of teachers in California. They also found a higher risk for ER-negative, PR-negative breast cancer in relation to cadmium and arsenic. So cadmium, lead, and mercury were the metals that showed up as these results. And it, was interesting to see this. We know that in particular for the evidence for cadmium is strongest compared to the other metals, that it has these endocrine disrupting capabilities. And also all three of these metals, suggest a role for industrial emissions. They all have common sources, including coal burning, municipal waste incineration, and metal processing. But a limitation of this study is that we're relying on this database, the NATA database, which really relies on industries to self-report their emissions. And there's no monitoring of these hazardous air toxics. And so that is a challenge when you're interested in these hazardous air pollutants. There's really not a better way to assess exposure, especially over such a large geographic scale, compared to the criteria air pollutants, which are much better monitored.
Yeah, Lexi,
So,
I'm also wondering, are there any validation studies for the exposure for the metals? And also, are there any studies find associations with other suspected outcomes of interest?
Yeah, so there have been some small validation studies, and they have found a modest correlation between I think it was actually specifically focused on some of the metals in relation to some local air monitors, but it is really challenging because what NATA estimates is an annual average. And so often you're comparing apples and oranges and you're comparing it to some short-term monitor that's really not getting at that. We have been working on trying to validate that with some biomarkers, and we do not see that the airborne metals are related to the metal biomarkers that I've measured.
Yeah, the biomarker may be transit, right? Maybe
well, we're looking at toenail metals, which we would hope would be a longer term measure, but it does, it has a similar issue in that it's also incorporating metals from diet and smoking and a lot of other sources. So it's hard to validate these things. And there are other studies looking at these in relation to other health outcomes. I'm not as on top of that literature, but it definitely has been used more widely than in breast cancer.
Thank you.
All right, so unless anybody has any other question about the airborne metals, I'm going to move to the criteria air pollutants. So most of my work has focused within the criteria air pollutant realm, and the benefit of these exposures and considering them is that they are much more regularly monitored by the EPA. So we have a lot better data to estimate an individual's exposure to the criteria air pollutants. And this includes air pollutants, including particulate matter, nitrogen dioxide, and ozone. And so for breast cancer, there have been quite a few studies that have evaluated air pollution in relation to breast cancer risk. And the findings are really inconclusive, and it really varies based on the pollutant measured. So studies that tend to consider markers of traffic, such as NO2, tend to more consistently observe a positive association, whereas there's been largely null associations for particulate matter. And so this figure comes from a meta-analysis that came out last year, evaluating PM2.5 in relation to breast cancer risk. And you can see the estimates are kind of all over the place, and the summary risk ratio is right around 1. But PM is an aggregate measure of air pollution that's solely based on particle size. And so it really doesn't tell you anything about what composes those particles. And there's a lot of geographic variability due to varying air pollution sources in what is making up this PM. So the aim of our study in the sister study was to evaluate the association between these main criteria pollutants, NO2, PM10, and PM2.5, but importantly with consideration of the PM2.5 component mixture. And this study had a similar design as the previous one I discussed with the airborne metals, where we focused on exposure at the sister study enrollment using their geocoded addresses, and then following women for breast cancer risk, but not updating. The air pollution is solely at the enrollment address. But for this exposure assessment, we have much better measures. And so this was done in collaboration with investigators at UW and Joel Kaufman's group, using a validated regionalized universal kriging model with spatial smoothing. And so what this model does is it incorporates a whole host of EPA monitoring data, as well as a large number of geographic covariates, including population density, green space, traffic, industrial emissions, And it uses this to predict an annual average at the enrollment address for these three pollutants. These models are limited to women who are living within the contiguous United States. So when we looked at the association between PM2.5, PM10, and NO2 with breast cancer risk, we first started by stratifying by census geographic region because we were concerned about this potential heterogeneity in air pollutants over the US. And we did see evidence of heterogeneity really across all three of these pollutants, but most strongly for PM2.5. So among women living in the Western US, we found that PM2.5 was significantly associated with invasive breast cancer risk. And so we were able to explore that finding in a little bit more detail by incorporating PM2.5 component data. So in the sister study, we have identified subgroups of women who have similar PM2.5 component profiles using a mixtures method called k-means covariate adaptive clustering. And so what this method did is it took information on 22 different PM2.5 component species and identified 7 groups of women in the sister study. And sorry, this map looks a little blurry, but you can see these are, each dot represents a sister study baseline address. And these women were classified into subgroups who have similar PM2.5 component profiles, which should suggest the sources that the major PM2.5 sources that they're exposed to. And so our question was whether these PM2.5 component profiles may modify the association between overall PM2.5 mass and breast cancer risk. And so when we stratified the association between PM2.5 and breast cancer by these clusters, we did see evidence of heterogeneity. And so we see a higher risk for breast cancer among women who were assigned a cluster 4. And these are the black dots that are in the California region. And so this cluster's profile was defined by being low in sulfur and high in sodium and nitrate, which was indicative of agricultural emissions in this area, as well as marine aerosols. We also saw a higher risk among women living in cluster 7, although it was more imprecise. And this is a more diffuse Western-based cluster. It's the gray dots that are kind of spread out across the West. And this profile was characterized by being high in transition elements, which is really thought to be indicative of the surface soil in the Western US. So this was the first study to consider how PM2.5 mixtures may impact the association between PM2.5 and breast cancer risk. And I think this study was really important given the fact that PM2.5 composition is so heterogeneous across the US. And it makes sense that previous studies that have just estimated associations with total PM2.5 mass, particularly over large geographic areas, may have masked over this relevant heterogeneity in associations and could explain the largely null findings that have been observed in the literature with breast cancer. So I wanted to explore this heterogeneity in another large US-wide population. So I'm going to make a little bit of a detour from the sister study and just talk briefly about a study I did in the Black Women's Health Study. And the Black Women's Health Study is another large US-wide cohort, but this population is limited to Black women. And this is an understudied population in breast cancer generally, but also in air pollution. And it's an important population because we know that Black women tend to live near more carcinogenic sources of air pollution, and they're also more likely to be diagnosed with more aggressive subtypes. And so in the Black Women's Health Study, we considered air pollution in relation to breast cancer risk among 41,000 participants, and they had about 2,000 breast cancer cases. And we did not see strong evidence that their baseline air pollution exposure was related to breast cancer risk. But again, when we stratified the association between PM2.5 and breast cancer risk by geographic region, because we do not have the same component data in this population, we saw, again, this significant heterogeneity for PM2.5. So among women in the Black Women's Health Study, we observed a higher overall breast cancer risk for PM2.5 among women living in the Midwestern US. And this association was even stronger when we considered ER-negative tumors. So taken together with our findings from the sister study, both of these populations really emphasize the importance of considering how PM2.5 heterogeneity may impact your findings. But obviously in the sister study and the Black Women's Health Study, we found different regions appearing to be more important. And these studies are very different in a number of ways, including that the Black Women's Health Study was about 10 years prior to the sister study. But they also had focused their air pollution exposure assessment only in metropolitan areas. So I think we're assessing very different air pollution exposures across these two cohorts. But another potentially important difference is that the sister study is a population of women who all have a family history of breast cancer. And so when I was trying to reconcile these findings, I wanted to explore this a little bit deeper. And so because of the sister study enrollment criteria, all of our participants do have this underlying familial risk of breast cancer. And it's possible that this may make women in our study more susceptible to environmental exposures. And one study has shown that a greater extent of breast cancer family history was associated with reduced expression of certain DNA repair genes. And this could be particularly relevant for breast cancer because we know that for some of the constituents of air pollution, they may be acting via DNA damage. And so this could modify an individual's response to exposure to air pollution. And so in a work led by my postdoc, Nicole Niehoff, we looked at the association between air pollution and breast cancer risk, stratifying by extent of family history. And in this study, we used the Bodicea measure, which is an algorithm that incorporates detailed information about a woman's family history to identify and quantify their underlying familial risk. And so we found that in women who had a very high Bodicea score, which was defined as greater than 90th percentile, among those women, NO2 was significantly associated with a 30% higher risk of breast cancer, whereas we did not observe that association among the low risk women. So these findings do suggest that the sister study also might be more susceptible to air pollution exposure. And so next, I'm going to shift gears a little bit and talk about some of the other work we've been doing on other novel carcinogenic attributes of PM2.5. And this is some recent work on radioactivity of fine particles. So radioactivity of fine particles is an understudied aspect of air pollution carcinogenicity. Radon is a naturally occurring source of ionizing radiation that varies geographically across the United States. So you can see this map shows radon levels across the US with the red shading indicating higher levels of exposure. And you can see that radon levels do tend to be higher in the Western US. And so particulate matter can actually be a vector for radioactive isotopes. Radon decay products can attach to PM2.5 particles. And so once these particles are inhaled and deposited in the lung, the progeny then can release radiation to surrounding tissues. And previous studies had shown that particle radioactivity was associated with biomarkers of oxidative stress and inflammation, but no study yet had considered associations with cancer. So I did this work in collaboration with investigators at Harvard who had developed a nationwide spatiotemporal ensemble model that estimated residential exposure to gross beta particle radioactivity. And this is a bit of a crude model. It's at a 32-kilometer grid, but when we applied this in the sister study, we saw that women who lived in areas of higher particle radioactivity did not have a higher risk of overall breast cancer or ER-positive tumors. But we did see this suggestive increase for ER-negative tumors. And we have fewer ER-negative tumors in this population, so our confidence intervals are a bit wider. But we do see this suggestive increase across all of the quintiles. And when we limited this to women who were residentially stable at baseline, meaning that they had reported living at their baseline address for more than 10 years, these findings were strengthened. And so this paper is currently in press at EHP. But these findings are really interesting, and I hope that we can apply this exposure model in other populations to confirm these findings. So I'm going to summarize the work that I've gone through today. I know that I went through quite a few studies. So first we looked at the airborne metals, and we found a suggestive association, which for cadmium, mercury, and lead, which suggests a potential role for industrial emissions. I've also done a number of studies evaluating PM2.5, and these studies do support that PM2.5 may play a role in breast cancer risk, but particularly when considering geographic heterogeneity and exposure. Our findings also suggest that women with a breast cancer family history may be particularly susceptible to air pollution exposure, as well as a possible role for particle radioactivity in ER-negative breast cancer risk. But despite my work in this area, as well as others that are increasingly suggesting that air pollution is related to breast cancer risk, we really don't know much about potentially relevant biologic pathways. And so I've done some work incorporating breast tissue characteristics as a plausible intermediate outcome. So for the last part of my talk today, I wanted to share some of the work we've done on air pollution and breast tissue characteristics in the Komen tissue bank. And so this work has focused largely on terminal duct lobular units, or TDLUs. And TDLUs are the epithelial structures, which are the primary site where most breast tumors arise. So you can see in this image, there are 4 TDLUs that their span is marked by the red lines. And so as women age, TDLUs involute or shrink. And so having a lack of involution, meaning you have more TDLUs for your age, is associated with a high risk of breast cancer, which makes sense because there's more at-risk epithelium for more sites for breast cancer to arise. And so this is a plausible intermediate outcome for breast cancer. Previous studies have found that the established reproductive history, breast cancer risk factors, as well as estrogen levels are associated with this marker, but really little is known about how environmental exposures may be associated with TDLUs. And so this led me to collaborate with the Komen Tissue Bank, which is a really unique resource. It is a biorepository of about 5,000 women who have donated normal breast tissue samples, which is pretty amazing. And within the Komen Tissue Bank, they have a subset of women of about 2,000 women who have existing TDLU measures, which were ascertained from a trained pathologist. So you can see the Komen population is the average age of donation was about 41 years. So they're a bit younger than the sister study. And it's also a slightly more diverse population with 19% non-Hispanic Black and 9% Asian Hispanic or other. So we took the women's residential address at the time that they donated their normal breast tissue, and we linked it with the EPA CMAC modeling system to estimate their residential exposure to PM2.5 total mass, as well as PM2.5 components. And so what we found is that living in an area of higher exposure to PM2.5 was associated with higher TDLU counts. For example, an IQR increase in PM2.5 was associated with 11% higher TDLU counts among postmenopausal women. And so this was consistent with our hypothesis that air pollution, and especially exposure earlier in life, may be impacting the normal histologic characteristics of the breast, which may predispose it to being more likely to develop breast cancer later in life. And so we're currently working on expanding these findings into the full Komen tissue bank cohort. And to do that, we're collaborating with investigators at NCI and Gretchen Gerich's group, who are using a machine learning algorithm and applying it to all the biopsies in the Komen tissue bank. And so what this algorithm does is it takes the tissue section and it quantifies the epithelial, stromal, adipose, and total tissue area in an automated and reproducible way. So you can see in the slide, in the picture, slide A is what the biopsy looks like before the algorithm, and then in the panel B, you can see that the regions have been shaded in order to quantify these different parts of the tissue. And so the investigators that we're collaborating with have previously used this method in a cohort of women with benign breast disease and found that women who have a higher percent epithelial area and a higher epithelial to stroma proportion, or ESP, have a higher risk of breast cancer later on. And so currently we're working on evaluating associations between PM2.5 and these histologic characteristics among the Komen population. And what we see is among women in the Midwestern US that exposure to PM2.5 is associated with higher percent epithelium and higher ESP, but not percent stroma or percent adipose. So we are seeing associations with these two characteristics of the breast tissue that have been previously found to be related to breast cancer risk. But this is really only evident among the Komen tissue bank participants in the Midwest. And we see substantial heterogeneity by region, and these results are really not evident among women living in other regions. And the Komen tissue bank started at Indiana University. And so really a lot of the participants are concentrated in the Midwestern US, and they've had sites now outside of it, but we really don't have the power to investigate these other regions very well. But I think this is another example really demonstrating how heterogeneous PM2.5 is and how this, may really play a role in breast cancer risk. So we're currently continuing to explore PM2.5 component patterns among these women and trying to elucidate what PM2.5 components might be driving these associations. So I'm going to end by just talking about some of my future directions in air pollution and breast cancer. So a lot of the limitations of the prior work that I've talked about even today is that even in a cohort as big as the sister study with 50,000 women, we still run into issues of limited power, particularly when we want to consider relevant subgroups such as by tumor subtype or focusing on premenopausal women. And as Hung Lee brought up very astutely early on, we often really just consider a single adult address as a proxy for long-term exposure, which we know is, potentially not the most accurate exposure assessment. So I'm working now on a pooled study that is pooling data across 5 different large cohorts, so the Nurses Health Study 1 and 2, the WHI clinical trial, and the Women's Health Observational Study in addition to the sister study. And so this is building off of an existing R01 led by Joel Kaufman, and we'll be using his newly developed air pollution exposure models, which have improved temporal and spatial resolution. But really, the strength of the study will be that we have almost 500,000 women across these cohorts. And so I think we'll be able to really consider how associations vary by subtype, in addition to menopausal status, geographic region, and race ethnicity. And race ethnicity is really important because I think that this has been highlighted very well recently in a report by Tessem et al. That came out last year, that in the United States, Black people are exposed to greater than average exposure to PM2.5 from all sources. And this was also highlighted in the New York Times. And so hopefully we'll be able to address those disparities in our pooled study. But we're also going to be doing some more work incorporating and trying to understand industrial emissions. So for this project, focused on industrial emissions, we'll be doing work using the EPA toxics release inventory, which will allow us to estimate residential exposure to point sources of industrial emissions in the sister study. And the goal of this project is to evaluate overall carcinogenic emissions. So using a spatial-based mixtures approach to estimate if you're exposed to multiple point sources simultaneously, what is the impact of that on your overall breast cancer risk? And the other goal of this project is to consider disparities in exposure by both individual and neighborhood level characteristics. Some of you may have seen this report. It got quite a bit of news late last year, and it was led by ProPublica, and they created a detailed map of cancer-causing industrial air pollution in the US. And as part of their report, they noted that there are these existing EPA risk estimates that really lack consideration of these impact of multiple exposures, as well as issues of environmental justice. And so hopefully those are 2 issues that we'll be able to address in this study. And finally, the last project I'm going to share with you guys today is some work we're doing trying to get at historical air pollution exposure. So in the sister study, we have information on the enrollment address, which is what most of my studies have focused on. But we also asked women about their longest lived adult address. And moving forward in the sister study, we have these their follow-up addresses, which we'll be able to use in the pooled study and looking at time-varying air pollution exposure moving forward from enrollment. But we do have these missing addresses, and they're different for every individual, depending on when was their longest lived adult address. And so we're working on filling those in, and we're doing that in collaboration with LexisNexis, which is a commercial database where you can get individuals' address histories going back in time. So we are going back to 1980, 1990, depending on the quality of the data. And so the goal, and we're also conducting a validation study. So as part of this, we will ask 1,000 participants in the sister study to confirm these residential histories that we build for them. But the goal of this study is to really assess historic exposure to PM2.5 and NO2. And this, I think, will really get at an important limitation in the literature, is that we often have this somewhat arbitrary adult exposure, which just happens to coincide with when we enrolled people in a study. Whereas for breast cancer in particular, we know that exposure decades prior is probably very relevant, particularly during potential windows where the breast is more susceptible, such as the woman's reproductive years or the menopausal transition. And so these are considered potential biological windows of susceptibility. And so hopefully with this now new resource of having these residential histories, we'll be able to both get at historic exposure, but also at these potentially a priori selected windows of time that may be more relevant. I just want to end with some opportunities for collaboration. I know that we have missed the deadline this year, but we do have a really great summer internship program that's out of NIH. And because of COVID, some of the silver linings of COVID is that it is hybrid. So there is a virtual component. So I would encourage people who might be interested to apply next year. It's a great way to do a research project. Most of our summer fellows end up publishing their papers from the summer. There's also opportunities for dissertation research within the sister study, as well as pre and postdoc positions. And I am actually currently recruiting for a postdoc. So if you know anyone who is interested in the intersection between environment and cancer, please have them contact me. My email is here. And with that, I want to thank my amazing collaborators and sister study participants. And I'd be happy to take any questions.